day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Dita Omakwala. This program is brought to you by the Northern Cape Department of Education, and it is broadcast by Pagama Research and Development. So in today's lesson, we are looking at our Term 1 revision, and we are looking at DNA, the code of life. So here we will focus on DNA replication as well as DNA profiling. So we're going to look at the process whereby DNA makes an exact copy of itself, and then we will then look at DNA profiling, whereby we are going to be looking at how a DNA profile is made, as well as how to interpret a DNA profile and the uses of these DNA profiles. Okay, so by the end of our lesson today, you should be able to describe the process of DNA replication. You should be able to interpret a DNA profile, and you should also be able to describe the uses of a DNA profile. So we're going to look at the process of DNA replication, how to use a DNA, what a DNA profile will be used for, and how to interpret it. So we're going to look at a DNA profile and just get to understand what kind of information that DNA profile gives us. Right, so our pre-knowledge would still be the structure of DNA as well as RNA. All right, and our baseline assessment before we get to our lesson today, we have two questions. The first one says label parts 1, 2, and 3. So our part 1 here, because we are given thymine here, it means that we know that this would also be a thymine because we're looking at the shape. So that means that part 1 would be adenine, and then part 2 would be a deoxyribose sugar, and then part 3 would be a weak hydrogen bond. All right, so it is an adenine, a deoxyribose sugar, and weak hydrogen bond. So remember, if you are given a guideline, you should then follow that and be specific with your answer when it comes to the nitrogenous bases. Second question, it says how many nucleotides are shown in the diagram. So remember, to count your nucleotides, you can simply count your nitrogenous base. You can also just count your sugars or your phosphates because each nucleotide is made up of one of those. So let's count our nitrogenous bases. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it means that we have 10 nucleotides in this case. All right, our terminologies, we have DNA replication. This is the process by which a DNA molecule replicates to produce an exact copy of itself, to make an exact copy of itself. The cell cycle, that is a series of events that occur during the lifespan of a cell. So we look at the cell cycle when we look at processes such as meiosis and mitosis. Template, that is a strand of DNA upon which another strand of DNA is built during replication. So we're going to look at that now. And then DNA profile, those are the black bars that are left behind on X-ray film when an extract of DNA undergoes a biochemical process. Lastly, DNA profiling. This is a process of identifying an individual by comparing his DNA with that of another known DNA. Now, as we are looking at DNA profiling and a DNA profile, you should be able to identify these and differentiate between these two. So DNA profiling is a process, and DNA profile are the black bars that are used during the process of DNA profiling. So you should be able differentiate between those two. All right. So as we have said, DNA replication is a process whereby DNA makes an exact copy of itself during DNA replication, right? Where and when does it take place? It takes place before cell division begins. So it takes place before mitosis or meiosis can take place, because it takes place in preparation for these two processes. It needs to take place before they can take place, right? So it occurs before meiosis, and meiosis can take place, right, in the nucleus, and also it takes place during interphase. So we know that there's interphase before both mitosis and meiosis can take place, right? Okay, this process takes place in the nucleus of the cell, right? Okay, and it is carried out in preparation of cell division, which, as we have said, that is meiosis and mitosis. All right, Now, why is it important? What is the importance of DNA replication? So we know that this is where the DNA makes a copy of itself, like, isn't it? An identical copy of itself. Right. So the DNA that we would have at the end, the replicated DNA that we would have at the end of this process, will be a DNA that contains two strands. One original strand. So the one original strand will be the strand from the DNA that we started off the process with, and a newly made strand. So one new strand and one old strand. As we can see here, there is the new strand. Let's have a look. So we can see that our old strand is blue in color, right? So this would still form part of our old strand, right? So let me just use the correct colors so we can see that here our DNA, our old DNA is blue in color, as we can see over there. That is our old DNA. It starts from here. So this is where the old DNA starts, right? And then as DNA replication takes place, we are going to talk about what happens exactly. It's going to take place. As it takes place, it is going to form a new DNA strand. Now that new DNA strand is the one that we can see in red here. So there's the red together with the blue, just to identify which one is the old and which one is the new. Now, this results in identical copies of the DNA within the cell. So now we're going to have two copies, which is pretty much the same as when you make a copy. When you place a paper into a photocopying machine and you come out with two papers that are identical, that is simply what happens during DNA replication. But we're going to look at the steps and exactly how it happens. So why is it important? It is important because it makes sure that the daughter cells that result from mitosis each contain identical genetic material. So remember, during mitosis, we end up with two cells that are exactly the same, right? So for these two cells to be exactly the same, especially if they're only made up from one cell, it means that from that one cell, there should be enough material to be divided amongst the new cells and the old cell, right? Because the genetic material has to be the same. So DNA replicates, the DNA doubles so that there can be enough genetic material to be shared amongst these two new cells, and for that genetic material to be exactly the same, for those two cells to be the same. Because remember, mitosis results in two cells that are identical to each other, right? So that is that the DNA that was present in the parent cell should also then be found in the new cell. As we can see here, we start off with this, right? And we are going to end up with two cells that have exactly that. Now, if we started off with this and there wasn't any DNA replication to result in this cell, it would mean that we will not be able to have the two daughter cells that are identical. So DNA replication takes place so that the genetic material can double and that genetic material can be shared amongst the two cells equally, right? So the replication of DNA also makes sure that the two new daughter cells can contain the same amount of DNA. So the number of chromosomes, that is actually the main thing that we talk about when we talk about identical cells. We talk about the fact that if you start off with 4 chromosomes during mitosis, the two new cells will also have to have 4 chromosomes. So the amount of DNA of the daughter cells should be the same of the DNA of the parent cell. Then with replication during meiosis, remember we have two divisions here. We have meiosis 1 and meiosis 2. So one round of the DNA replication is followed by 2 rounds of cell division, meiosis 1 and meiosis 2. And those 2 rounds will then result in 4 haploid cells. Now, in this case, we only have haploid cells. We do not have cells that are identical to the parent, right? So that crossing over of the DNA will result in exchange of genetic material, right? So that also, those cells need to be shared properly so that at the end we will be able to have the haploid number of chromosomes within those cells that we are going to result in. Now let's have a look at the process of DNA replication. What exactly happens during DNA replication? Right. So first you would start off with a twisted DNA molecule, right? So that DNA molecule, the double helix, will have to unwind. When it unwinds, it is going to now form this ladder-like structure that we are looking at here, right? So we have that ladder-like structure that we can see here. And then secondly, the weak hydrogen bonds will have to break. So the weak hydrogen bonds that are holding the nitrogenous bases together will break, which is what we are seeing here. We can see that the weak hydrogen bonds have broken here, and the two strands are starting to separate, right? So the unwound DNA strand will now serve as a template. So we say that this is now a template. So this part is a template, and this part is also a template. They are used as templates for the formation of the new strands that are going to be formed, right? So now we have our template, and then we are going to have free nucleotides with matching nitrogenous bases that are going to line up next to the exposed bases in the original strand. So the templates are the original strands, or we can say the original strands are these templates. So now we spoke about free nucleotides that are found in the nucleoplasm in the previous lesson. So this is what we were referring to. So these are the free nucleotides. You can see that they're not attached to anything. So they are free nucleotides, and these free nucleotides will attach themselves to the complementary bases. That is what we would be referring to when we talk about complementary base pairing. In this case, we mean that thymine will be pairing with adenine and guanine will be pairing with cytosine. So as we can see, here we have an adenine, right? So it means that this adenine will pair with the thymine, and the thymine that was pairing with that adenine will also pair with an adenine, right? Just like here again, just like that. Then we are now starting to have two strands of DNA that are being formed from those two old strands of DNA which are serving as templates, right? So now the new deoxyribose and the phosphate molecules join to form a chain alongside each of the strand of the old helix. So now the sugar and the phosphate will be joining together with these nitrogenous bases that are forming complementary base pairs with the old nitrogenous bases, and now we are going to start seeing the formation of the new DNA molecule, right? So the hydrogen bonds will rejoin or will reform to join the two matching strands of the new molecules with each other, right? And then we are now going to say that we have two identical DNA molecules that are called the daughter strands that are formed, right? Now, each double strand will rewind to form that new helix, and the new double helix will wind itself around what we call the histones, right? Okay, let's just have a look at that again. So as we can see now here, this is our old strand, and this is our old strand, right? And then over here, these two would be our new strands. We can see that based on that. So this is the G that was paired to the C. Now the C is pairing to another G, and this G is pairing to another C, right? And that will happen going downwards. Wherever there's a G, it will pair with a C. Wherever there's a C, it will pair with a G. Wherever there's a T with an A with a T, until we end up with these two strands. So now we have two identical DNA molecules, as we can see, CG, GC, GC, AT, AT, as we can see all the way to the bottom, they are exactly the same as each other, right? Each DNA molecule here is made up of one old strand and one new strand. So this would be our old strand, this would be our old strand, this would be our new strand, and this would be another new strand, right? As you can see here, it started off as a double helix. The double helix unwinded. As it unwinded, it was now a ladder-like structure. Then it unzipped. As it unzipped, it also the weak hydrogen broke and it unzipped. That allowed the free nucleotides to pair with the complementary bases. As it paired with the complementary bases, the sugar and the phosphate formed the sugar phosphate bonds, and that then formed the backbone. And then that is when we can see that we are starting to have the new DNA, and that DNA will wind again to form a double helix structure. Right. Okay. As we can see here, now the old strand, there we have our old strand, and then we have our new strand. So as I've said, each DNA molecule will have one old strand and one new strand. Here we'll identify the old strand by the color blue and the new strand by the color red. All right, let's have a look at an activity. We have activity 2.1. It says the diagram below represents DNA replication. Okay, let's have a look at the parts that were not labeled. So we can see that here we have X and Y. So Y would be our sugar because it is bonded to the thymine. X will be the phosphate. So because Y is bonded to the thymine, it means that V is the adenine. And then molecule W would be our nucleotide, right? And then U would be our DNA molecule. And then Z would be the weak hydrogen bond, right? Okay, now let's have a look at the questions. 2.1.1, it says identify molecules W and U. So W is nucleotide, U is a DNA, right? Okay. And then B, it says identify parts of the molecules W, which are X and Y. Let's have a look at X and Y. X and Y is the phosphate and the deoxyribose sugar. We can say it's a deoxyribose sugar because we are looking at a DNA molecule, right? And then bond Z would be a weak hydrogen bond. Okay, 2.1.2 says where in the cell does the process occur? So we know that this process takes place in the nucleus. And then it says, name the phase of the cell cycle during which replication takes place. So remember, DNA replication takes place before mitosis or meiosis can take place, and it takes place during interphase. So that phase is called interphase. And then 2.1.4 says, explain the consequences if the type of bond Z is not able to break. So what would happen if the weak hydrogen bonds do not break? It means that the strand will not be able to unzip or separate, right? And then that will mean that the DNA will not be able to replicate because the free nucleotides will not be able to bond to any exposed nitrogenous bases. And therefore, it also means that protein synthesis will not be able to take place. Because for protein synthesis to take place also, it means that the DNA has to open up in order to form RNA as well. The mRNA is formed in a similar process to the process of DNA replication. So without that, those weak hydrogen bonds breaking, it will not be possible for protein synthesis to take place as well. Okay, now let's have a look at DNA profiling. So when we talk about DNA profiling, we will be talking about the profiling or a process of profiling whereby DNA of an individual will be used in order to form what we call a DNA profile, and that DNA profile will be used to check for some similarities between the DNA profile that they know of and the DNA profile of the people that they want to check. So before we go to that, let's have a look at fingerprints. So the fingerprints that we have on our fingers, right, as you can see over here. So everyone has their own set of fingerprints, right? Except for identical twins. Identical twins have identical DNA, they have identical fingerprints and features, right? So the fingerprints have been used since 1930 to identify suspects in crime. So we are still using fingerprints to identify individuals where possible, right? So there are some problems with these fingerprints, so they cannot always be used successfully because they can only be found on the tips of our fingers. This means that should anything happen to the tips of my fingers, it means that I will not be able to be identified using fingerprints, as they can easily be tampered with, right? Also, these fingerprints can be altered by the use of plastic surgery, meaning that I can also alter these should my fingerprints be needed and I do not want to produce that, right? Therefore, they are not always reliable in identifying criminals, right? Because they can alter them. Also, individuals would cover their fingerprints when they are trying to commit some kind of crime. So they can wear gloves. By wearing gloves, it means that the surfaces that they would have touched will not leave their fingerprints. So that is another thing that can be problematic with fingerprints, which brings us to DNA profiles, right? So now with DNA profiles, we know that the DNA that is found in every cell of an individual's body is identical, right? So in a person's body, every cell has this DNA, unlike with the fingerprints that are only found in your fingertips. Now, if they want my DNA, they can find DNA in almost every part of my body, right? So this DNA is easy to find, right? The DNA of every cell is identical to each other. Why is it identical to each other? Because it was produced by mitosis, right? So you cannot alter the DNA. If you leave a blood stain at a crime scene, that DNA will be the same as any other DNA in your body, including your saliva, including a small cell that can be taken from your body, right? So we find that the DNA is more useful in identifying individuals as compared to the use of the DNA fingerprints, right? So what are these DNA profiles basically? So remember I said you should be able to differentiate between a profile and profiling, because a profile is what we are looking at here. This is a DNA profile, right? So this DNA profile contains black bars that are left behind on an X-ray film when an extract of DNA will undergo a biochemical process. So a DNA will be mixed with certain chemicals and placed into this X-ray film, and then the DNA will run through that X-ray film, and then it will leave these bars, right? Now, because your DNA is exactly the same, if a DNA profile is made and it looks like this, another DNA profile can be made the next day from a different part of your body. It will also still look like this one that we have here, because the DNA within your body is always the same, and it will always produce a profile that is the same. However, when we produce a DNA profile from a different individual, it will not look like this, because their DNA is not the same as yours. The proteins, everything within their DNA will not be like this. So this is a DNA profile, and the process of producing this and the technique of producing this and using this profile is what we call DNA profiling, right? So these profiles are made using a sample of DNA from the body, tissues, or fluid. Like I said, it can be blood, it can be saliva, it can be parts of the body. A cell from the body can produce these DNA profiles, right? So from your hair, from your blood, the skin, as well as saliva, all of these can be used to produce these DNA profiles. So that sample that is taken from the body is going to be broken down into smaller pieces. As it is broken down into smaller pieces, the DNA is exposed and it is then marked with some radioactive strobes. So it is going to be mixed in order to mark it so that it can be able to leave those black bands that we have seen in the previous slide. So that's sample will be taken. It will be, then the DNA will be extracted by breaking it into pieces. It is then going to be mixed with the correct it is going to be mixed with the correct strobes, and then it will be placed in that X-ray, and this is what we will be left with at the end of the whole process, right? Okay, now if this is from the same person, it means that the profile will be the same. If it is not from the same person, the profile will then look different. So the pattern of black bars are going to be formed, and if one DNA profile is compared to another and they match, we know that it is then from the same person. Now let's have a look at how to interpret these DNA profiles, right? So remember, it is a process of identifying an individual by comparing their DNA with that is known, a DNA profile that is known. That is the DNA profiling using a DNA profile. So when you go through questions like this, it's very important to use a ruler so that you can be sure that the DNA profiles are, the bars on the DNA profiles are actually aligning with each other, right? So what you would do here, let's have a look at this example, right? So this is done by comparing the bands on the DNA profile of an individual to those of a DNA profile from a crime scene or another individual. So we can do this in paternity testing to compare with the mother and the father to see if they are, the mother and the child and the father to compare if the parents are the parents of that child, or to check if a certain person has actually committed the crime. So in this case, we're looking at at a crime scene, and there's a victim. So we have the sample of a victim, right? And then we have the sample that is coming from the crime scene. So because this sample does not look like that of the victim, it could then mean that it is the sample of the suspect, right? Now, we have three suspects. These three suspects are then required to produce their DNA samples so that they can be checked, and their DNA profiles can be produced in order to compare with this DNA profile from the crime scene, right? Now, we are going to compare the black bars or the black bands to of the suspects to the one from the crime scene, right? So let's start with this one. So now if we align this one, we would see that it matches with suspect 2. So you're going to place a dot on the bar of the suspects that matches with the one from the crime scene. So suspect 2 now has one bar matching, whereas the other suspects do not have any bars matching. We check the second one. The second one matches suspect 1 and suspect 2. So you place a dot on suspect 1 and the second dot on suspect 2. This still puts suspect 2 as the big suspect because he has 2 bars matching. The third one, suspect 1, suspect 2, and suspect 3. So now you place dots on both of them, all of them. Now, still suspect 3 2 is leading because he has 3 bars matching, and then the fourth bar only matches suspect 2. Bar number 5 matches suspect 2 and 1. It doesn't match with 3. So you put the dots, and then the last bar matches both 1 and 2. So suspect 2 has all the bars matching. That means that suspect 2 is the one that committed the crime because the sample that was found on the crime scene matches with his DNA sample. Okay, now let's have a look at what exactly can these DNA profiles be used for. Already we know that it can be used at a crime scene. We know that it can be used for paternity testing, right? So it can also be used to diagnose some genetic disorders. If it is used to do that, it can also then be used to develop some cures for these genetic disorders, right? And then it can be used as biological evidence. When we're talking about biological evidence, we are talking about being used at a crime scene to solve certain crimes, And it can also then be used to identify relatives. That means paternity testing. It means testing between siblings or other relatives. So it doesn't really mean that it can only be used for paternity testing, but it can also be used to check if people are related. So when we are talking about identification of relatives, it can also mean identification of people who are deceased. So we can say that maybe a person has been burnt beyond recognition, right? So that means that if there are some remains of that person's body, they can take the DNA sample and they can take the members of the families of the people who suspect that the deceased could be their family member to compare. So now they would take the samples of the different individuals from different families together with the sample of the deceased individual. Now they're going to compare them the same way I compared the crime scene with the suspects. Now, whoever matches most with the deceased would then be the one that is said to be the relative of the deceased, right? Now, in this case, they will not match exactly like with suspect 2 and the crime scene, because remember, the family member and this deceased are not twins. It means that their DNA is not exactly the same. However, because they are related, maybe they are siblings, it means that they do share some DNA because they come from the same parents. So there is going to be some degree of similarities, which would then tell us that they are related. Okay, so with diagnosing of inherited disorders, the DNA profile is used for that in case of diseases such as hemophilia as well as sickle cell anemia, and the diseases may be diagnosed in newborn babies as well as in unborn babies. So this can be done in newborn babies and also in unborn babies, right? Okay, and then we have the development of cures for the inherited disorders. So these DNA profiles will help with research of the further research of the inherited disorders. So the researchers can use those profiles to find patterns in large groups of people with and without the disorder. So the more they understand the disorder, the more they will be able to find the necessary cures for that disorder. Right. And with the biological evidence, like I said, it can be in forensic sciences in order to find criminals. It has now become more reliable than the use of fingerprints, as we have said. So this is a reliable method of putting criminals behind bars. However, that does not tell us whether the individual has committed the crime. It only tells us whether the person was present in the crime scene or not, meaning that there has to be more evidence to prove that this person has actually committed the crime or not. Right? Okay. Lastly, with the identification of relatives, as I've said, it can be used to determine the father of the child, which is paternity testing, right? So it can also be used to trace siblings that were separated at birth, and then it can also be used to identify people who have died and could not be identified, right? Okay, so as we have looked at that, and then now we're just going to look at how it will be used in paternity testing. So we can see that here we have the mother, we have the child, we have dead 1, dead 2, and dead 3. So this would be the 3 males who suspect that this child could be theirs, right? So now of these 3 males, we are going to compare their DNA profiles with the DNA profile of the child in order to see which of the males is the father of the child. Right. Now, this is done a bit differently from checking crime scenes. Now, here we have the mother. Now, remember, from our knowledge of genetics and inheritance, we know that every individual contains genetic material from their mother and from their father, right? So it means that the DNA that we see on the DNA profile of this child is a combination of the DNA of the mother and the DNA of the father, meaning that before we even compare the fathers, we need to first compare this profile with that of the mother, so that whatever bands match with the mother are going to be excluded when we match with the father, because this DNA profile needs to contain DNA from the mother and DNA from the father. So we are going to start by comparing the child and the mother. So the first band of the mother does not match with the child. Then the second band of the mother matches with the second band of the child. So I'm just going to mark both of them to show that these two bands are matching with the child. Third and fourth band of the mother do not match. And then the next band of the mother matches with the third band of the child. The last band of the mother matches with the last band of the child. So we have three bands from the mother matching with three bands of the child. Right. Now, we are going to focus on only the last three bands of the child that did not match with the mother. Then, if those three bands that are remaining of the child match with any of the three bands from one of these males, then that will be the father of this child. Let's start with the first band of the child. We focus on the band of the child because the males in this case have more bands. So the first band of the child, remember, make use of a ruler so that you can get proper results. So we can say that the first band of the child matches with dead 2 and dead 3. And then we skip the second one and the third one. We move on to the fourth one. It matches dead 1 and dead 3. So far we can see that chances are that dead 3 is the father, because dead 1 only has 1, dead 2 only has 1, and dead 3 has 2. Right. And then the third one, the second last one from the child, it matches with dead 2 and dead 3. So we can then say that the father of this child is dead 3. Okay, let's have a look at an activity. This is activity 2. It says study the diagram below, which shows the following DNA profiles. Remember, we do not call them genetic fingerprints anymore. We call them DNA profiles. And remember, this is a DNA profile. The technique used is DNA profiling. So here we have blood of a raped female victim. We have blood of 3 samples. And we have semen found on the female victim. So this semen from the female victim tells us that whoever the semen matches with is the person who raped the female. Now, earlier on, we spoke about the fact that the samples being found in the crime scene does not necessarily mean the person committed the crime. However, in this case, you cannot be found, your DNA cannot be found on the female victim if you did not commit the rape. So in this case, it would definitely mean that whoever DNA sample matches with the semen found in the female would be the one that committed the rape. So we have DNA from the female and we have DNA from the suspects. So we are going to compare the samples from the suspects and the sample of the semen, right? So if we have a look at the first one, it matches with the victim. It doesn't necessarily match with 3 because there's that gap over there. And then the second one matches with sample with suspect 3. The third one again with sample 3. The fourth one, suspect 3, again some suspect 3. So we can already see that this is matching with suspect 3. And then the last one again is suspect 3. So we can already see that it is suspect 3 who committed the crime. So which suspect was most likely the rapist? So it is suspect 3. Right. Explain your answer. So we can see that the DNA profile of the semen and the DNA profile that was found on the female, the semen that was found on the female and the profile of the male, 3 are matching. Therefore, that is why we say that he is most likely the one who committed the rape. Right. And then 7.3 says, give one reason why this evidence may be considered reliable. So why is this considered reliable? Why can we say that this is the one that committed the crime? So because of the way that the two profiles match with each other, we can then say that this is the person that committed the crime, because no one else can have this kind of DNA profile except if he had an identical twin. So if he does not have an identical sibling or an identical twin, no one else can contain that kind of a DNA profile that matches with his. So that is why we can say that he is the one that committed the crime, and we can say this is reliable information. Okay, 7.4, give two reasons why this evidence may not be considered reliable. So why can we say that this is not reliable? So there are some things that can cause this to be unreliable. Now, these profiles are done by humans in the lab, right? So you can find that maybe there was some mixing up in the labeling of the DNA profiles. They may have said the DNA profile of suspect 3 belongs to suspect 2 and suspect 2 belongs to suspect 3. So now, simply because it was labeled to be that of suspect 3, now suspect 3 is falling for a crime that he did not commit. So human error in the laboratory can lead to some mistakes. Deliberate swapping of specimen in the laboratory could be one of the reasons why we can say it is not reliable. And also, this requires a large length of DNA to get an accurate profile. So by simply taking a small portion, that can lead to some inconsistencies. However, if a bigger sample was taken, then we could say that the results are more reliable. And then lastly, name two benefits of DNA profile other than for solving crimes. So those are the ones that we spoke about, such as the diagnosing of genetic disorders, paternity testing, identifying dead persons. We can also have DNA research into variation of populations. We have spoken about variation of populations, whereby we look at variation within a species, right? And then also tracking individuals in populations is one of the other things that this can be used for, right? Okay, now we have activity 3. It says the victim of an attack scratched her attacker. A sample of blood and skin cells were collected from under the victim's nails. So it means that when the victim scratched this attacker, the sample remained. So this sample was used to produce the attacker's DNA profile. So that is the sample from the nails, which is that of the attacker, right? The DNA profiles for the victim and 3 suspects were also produced. The DNA profiles are shown in the diagram below. So we have the DNA profiles of the victim, the crime scene, and the suspects of the crime. Now, it says which suspect attacked the victim. Let's have a look. So the first one matches with 2 and 3. Second one matches with all of them. The third one matches with all of them again. And then the fourth one, it matches with 2 and 3. So far we can see that 2 and 3 are very similar. Then this one matches with all of them. But I think so far we can exclude suspect 1 because we've had 2 bands already that are not matching with the crime scene. And then the next one, it matches with 2 only, and it doesn't match with 3. Now that leaves 2 as the most definite one. Let's move on. It matches all of them. It matches 1 and 2. Again, it leaves 3 out, but 2 is still there. And then this one doesn't match 3. It's a bit thicker. It matches all of them. Matches 2 only. It matches all of them. So now that makes suspect 2 the attacker of this person, right? Okay. B, it says give an explanation for your answer. So for this explanation, you talk about the fact that all the bands on the DNA profile of suspect 2 match with the bands or the black bands on the DNA profile that was found in the victim's nails. So DNA profile for suspect 2 is identical to the DNA profile in the sample from the nails. 222. When confronted, the attacker says that the victim scratched herself in order to frame him. Explain why this is not possible. Now, if we have a look at this sample and we have a look at the sample of the victim, they do not match. And there's no way that the sample of suspect 2 could match with the suspect of the victim. So we can see from here, they are not matching. We can see here they're not matching by just simply looking at the bigger bands. So there is no way that this sample from the nails is the sample from the victim's DNA. So the victim's DNA profile is different from the DNA profiles from under the nails. Or you can say that the attacker's DNA was found on the nails and not that of the victim. Explain one reason why a DNA profile alone is not sufficient evidence to convict someone of the crime. So, like we said, this DNA profile just proves that this person was at the crime scene, right? So the DNA sample could have been planted under the nails, right, at the crime scene or under the nails. Like we're saying, the person could have been at the scene previously but was not involved in the crime. So you could have left your sample there, but you were not the one that committed the crime, right? Again, remember, we need large samples to confirm that this is actually reliable results. And some errors could have also taken place in order when the profiles was made, so that cannot be a reliable way. Last question 224. Explain one advantage of using DNA profiles in solving crimes. Right. So DNA profiles are unique. Only identical twins can have DNA profiles that are the same. So with everyone else, the DNA profile is different. So there's no way that you can alter this DNA profile or the DNA. So the DNA that you get, and if it matches, it means that it belongs to that person. So if your DNA is found at a crime scene, you must have been there. So it means that what's left now is to just prove that this person committed the crime as they were already found at the scene. So you cannot dispute ever being at the scene of the crime. However, you can argue that you did not commit the crime, but the DNA profile would have placed you at the scene, right? So it increases the chances of conviction, right? And it can then reduce crime cases. Okay, so remember we looked at DNA replication, whereby we looked at the fact that the double strand will unwind, and then that unwinding would then form a ladder-like structure, and then we would have the free nucleotides that would come in and bond with the old strands of DNA in order to form a new strand by pairing with the complementary bases. So through complementary bases, and at the end we'll have two DNA molecules that are going to be formed, which are identical to each other. Each strand will have one new strand, each DNA will have one new strand and one old strand. Right. Okay. And then in terms of DNA profiling, so this can be used for forensic sciences. It can be used for parentage testing. So with parentage testing, it cannot only be used for paternity testing. Remember, sometimes it can be the mother that wants to confirm that the child is theirs. Maybe the mother believes that the child that is presented to her at the hospital after birth is not hers, because maybe the child that she has seen immediately after birth does not look like the one that she is now seeing. So that can also be used to check if the child actually belongs to the mother. Medical diagnosis, as we have said, diagnosing of genetic disorders, as well as finding cures for those genetic disorders. It can also be used in the food industry as well as in agriculture. Okay, so in our next lesson, we are going to be looking at protein synthesis. So here with protein synthesis, we are going to look at two main parts of the process. We are going to look at transcription, which takes place in the nucleus, and we are also going to look at translation, which takes place in the cytoplasm. And then here we are going to look at how proteins are manufactured. Right. So here we have homework whereby it says study the DNA profile of 4 children given below. So we have these 4 children, and then we have the questions that you are going to study there. So I'm very interested in this question, which says describe how DNA profiling can be used for paternity tests. So remember, you would mention how this is used, whereby the mother is compared to the DNA profile of the child, and then the remaining bars would be the bars that are going to match with the bars of the father. Right. Okay, so we have come to the end of our lesson for today. Thank you for watching. I will see you next time. Goodbye.